This chart highlights a glaring contradiction in neuronutrition. On the left, observational data tracks a clear trend. Broad dietary patterns correlate strongly with preserved brain volume and a lower risk of dementia. On the right, we see the results of highly controlled clinical trials testing the specific nutrients found in that very diet. The data flatlines. If we look at large population studies, people who eat a traditional Mediterranean diet consistently show superior cognitive outcomes as they age. Yet, when researchers extract the active components of that diet, giving patients high-dose omega-3s or isolated B vitamin supplements in randomized control trials, the cognitive benefits virtually disappear. These failures suggest that clinical trials treat nutrition like pharmaceuticals, isolating single variables while ignoring the systemic biology of the human body. To decode how food actually protects the brain, we have to look past individual ingredients and map three interdependent biological systems: your energy baseline, metabolic buffering, and nutrient synergy. Resolving the cognitive diet paradox requires moving beyond single-variable biohacking to address these interconnected physiological states. Consider the MIND diet. It was developed at the Rush Memory and Aging Project specifically to prevent neurodegenerative delay by combining elements of the Mediterranean and DASH diets. A major trial published in the New England Journal of Medicine tested the MIND diet against standard caloric restriction. The results showed that both groups experienced similar cognitive benefits. Systemic energy availability dictates these outcomes. Cross-population data from the Bolivian Chiman hunter-gatherers, intermediate tribes, and industrialized societies illustrates the effect. Look at this graph plotting energy availability against physical brain volume. It forms a distinct U-shaped curve. On the far left, chronic caloric restriction results in a smaller brain volume. The body simply lacks the raw structural resources required to maintain neural tissue. On the far right, we see a similar drop. Chronic caloric excess actively degrades brain tissue and reduces overall volume. When dietary interventions succeed in average industrialized populations, it is largely because they pull patients out of chronic energy excess and restore metabolic homeostasis. That physiological correction overrides the nuances of specific nutrient combinations. There is a persistent assumption that cognitive health requires rigid, 24-hour micromanagement of specific nutrients to keep the brain fueled. Take omega-3 fatty acids. When you eat seafood, you are primarily consuming omega-3s in their triglyceride form. Your body doesn't demand immediate utilization of those lipids. Instead, your adipose tissue acts as a dynamic depot, sequestering these triglycerides for later use. During fasting or exercise, a process called lipolysis breaks down these stored fats, releasing the sequestered omega-3s back into the bloodstream. From the bloodstream, these fatty acids cross into the cerebrostyalbro fluid, providing the brain with a steady, continuous supply, entirely independent of what you ate that specific day. While specialized phospholipid supplements, like krill oil, can penetrate the brain faster in a single dose, continuous dietary intake normalizes in your tissue depot over time. Recent clinical data from USC tested this exact mechanism. They provided omega-3 supplements to patients over a long period, but because the subjects already had sufficient tissue depots, the intervention yielded zero additional MRI or cognitive benefits. Human physiology naturally buffers nutrient gaps. Running short-term, targeted trials on individuals who already have adequate nutrient stores guarantees a null result. This brings us to the most critical flaw in isolated clinical trials: biochemical codependency, or nutrient synergy. Consider homocysteine, a primary marker of B vitamin methylation status. While standard lab ranges might consider a level of 15 or 16 normal, cognitive risk actually elevates significantly once homocysteine climbs above 10 to 13. The VitaCog trial, run out of the University of Oxford, sought to test whether supplementing B vitamins to lower homocysteine could preserve cognitive function. The results revealed a strict interlocking dependency mapped on this 2x2 matrix comparing omega-3 status and homocysteine levels. With poor baseline omega-3s, lowering homocysteine yields zero clinical benefit. Conversely, supplying high-dose omega-3s provides zero cognitive benefit if homocysteine remains elevated. Supplementation only slowed cognitive decline when a patient possessed both high omega-3 status and normal homocysteine. This is a mandatory sequence of biological operations. B vitamin methylation and omega-3 integration rely entirely on each other to function. Testing isolated nutrients without first establishing a patient's baseline cofactor status ensures the trial will fail, masking the true clinical value of the nutrients involved. Cognitive health cannot be engineered by isolated superdoses. It requires maintaining systemic energy, utilizing your adipose buffer, and aligning your biochemical cofactors. We see this systems-based approach reflected in the Cosmos study, which tested the effects of a standard over-the-counter multivitamin in older adults. The data showed that the multivitamin only provided measurable cognitive benefits to individuals who started the trial with a poor baseline diet quality. These multivitamins provide a basic biochemical floor, ensuring the cofactors necessary for nutrient synergy are present. Instead of blindly taking supplements, the protocol is straightforward. Test your specific biological ranges. Check your vitamin D, your homocysteine, and your omega-3 levels to identify genuine deficiencies. By closing specific nutrient gaps and maintaining a neutral energy balance, we allow systemic physiology to preserve the brain.